The problem we'll be discussing today is with regards to high-fat feeding. Following a 48-hour high-fat diet, individuals showed an increased blood free fatty acid content and decrease in skeletal muscle membrane GLUT4 content. From the problem given to us, it is known that consuming a high-fat diet rapidly alters insulin sensitivity and glucose uptake in skeletal muscle. However, the mechanisms by which a high-fat diet alters insulin sensitivity and glucose uptake in skeletal muscles are unknown, presenting a knowledge void. Our purpose is to identify the mechanism that alters insulin sensitivity and glucose uptake in skeletal muscle following a high-fat diet. This information can provide insight on the effects of an acute high-fat diet on the insulin sensitivity and thus energy metabolism of a population. This is the overarching model that shows how glucose is taken up from the bloodstream by GLUT4 vesicles into skeletal muscle, liver, and adipose tissues. This process parallels itself in terms of free fatty acid entering each of the cell types via CD36. For this problem, we will only be focusing on skeletal muscle and adipose tissue. In our models, green arrows indicate activation and red arrows indicate inhibition. In terms of skeletal muscle, this is the normal way that glucose and FFA enter the cell. Insulin binds to the insulin receptor, and then through the signaling cascade, via PI3K, which activates RAC, and AKT, which removes the inhibition of S160, GLUT4 storage vesicles are mobilized to the membrane by RAC along actin filaments to bring glucose into the cell. AKT further activates CD36 translocation to the membrane, increasing conductance for FFA into the cell. Adipose tissue differs in that it has beta-adrenergic receptors and glucagon receptors that activate G protein to increase CAMP from ATP and then PKA. Inhibition of AKT results in less PDE, which is what inhibits the formation of PKA. Without PDE, we get an increase in FFA release into the blood from PL and HSL, extenuating the issue of FFA in the blood and decreasing insulin sensitivity. We speculate that insulin sensitivity decreases due to increased free fatty acids in the blood. As a result, this is going to increase free fatty acid entry into skeletal muscle. As a result, there will be an increased fatty acyl-CoA pool. Consequently, there will be an increased production of DAGs and ceramides. As a result of an increased pool of DAGs, there will be an increased inhibition of insulin signaling by increasing phosphorylation of inhibitory sites on IRS. Similarly, as a result of an increased pool of ceramides, there is going to be an inhibition of insulin signaling by increasing phosphorylation of inhibitory sites on AKT. As a result of decreased insulin signaling, there will be less RAC recruited. As a result of less AKT, there will be a decreased amount of AS160 phosphorylated. As a result of a decreased RAC activation and phosphorylated AS160, there is a decrease in GLUT4 translocation. As a result, there is decreased glucose transported through GLUT4 into the skeletal muscle. This increases the pool of blood glucose.
The study used to test our hypothesis investigated the effects of an acute high-fat diet on insulin sensitivity. The study consists of two conditions, a control group who consumed a regular diet and a group who consumed a high-fat diet over the course of 24 hours. They hypothesized that with an acute high-fat diet, there would be an increase in phosphorylation of inhibitory sites on insulin signaling cascade substrates in both skeletal muscle and adipocytes. The study used Western blotting to identify phosphorylation of inhibitory sites of AKT and also identified phosphorylation of inhibitory sites on IRS through Western blotting using anti-phosphatyrosine antibodies. They also measured membranous GLUT4 content via immunofluorescent labeling. The data shows that the increase in phosphorylation of inhibitory sites of both IRS and AKT in both skeletal muscle and adipose sites is associated with an increased pool of blood-free fatty acid. The increase in phosphorylation is also associated with a decrease in membranous GLUT4. This supports our hypothesis that increased intracellular free fatty acid increases the concentration of lipid-reactive species such as DAGs and ceramides, which phosphorylate and inhibit IRS and AKT respectively. The inhibition of these insulin signaling cascade substrates decreases insulin sensitivity via decreasing AKT activity, which is unable to phosphorylate AS160. This keeps GLUT4 storage vesicles anchored, decreasing membranous GLUT4 and conductance for glucose into the cell for a given insulin concentration.